So I think we're a couple minutes after, so I think we can get started. Welcome everyone to the first Biocurator working group call of 2023. I think this was mentioned in the December call, but we have rotated chairship of the Biocurator working group to both myself and Alicia Byrne from the Broad Geisinger grant for ClinGen, and Amanda is also doing coordination for us. And I wanted to thank Jenny Goldstein again for the wonderful job that she's done starting the group. I know she's here listening and passing over the leadership there. And we're excited to move on into 2023 and have some really good discussions with the biocurators and ClinGen. So today we're going to be talking about an overview of the ClinGen hearing loss gene curation expert panel and variant curation expert panel, and I will give this along with Andrea. And I'll just give a little bit of background about myself, and Andrea, if you want to give a little before you start, that would be great. So I'm a clinical molecular geneticist. I am the director of the ClinGen Biocuration Core based at the Broad Institute, and I'm also an associate lab director of the clinical research sequencing platform at the Broad Institute, involved in much of the gene and variant curation standard setting within ClinGen. And I've been with ClinGen for the last 7 or so years. So I'm excited to talk to you about one of my first expert panels that I worked with, the hearing loss expert panel. I'll turn it over to you, Andrea.
Okay, thanks, Marina. I'm Andrea Oza. I'm one of the biocurators for the ClinGen expert panels. I am a genetic counselor by training, and I'm a variant scientist in my current job. I've been doing curation for this group since we started way back in 2016. So yeah, I'm excited to share what we've been doing over the years and give updates on what our group has been up to. All right, I guess we can get going. Okay, so our clinical domain working group background is to create a comprehensive and standardized knowledge base of genes and variants that are relevant to both syndromic and non-syndromic hearing loss. So our goals were originally to identify and recruit experts in the field, and we wanted to get experts that represented the populations throughout the world so that we could have a diverse group of background and expertise for different populations. And then next was to develop, implement, and apply a variant assessment framework for hearing loss genes. And through that, we used the ACMG and AMP guidelines and specified them for hearing loss and deafness. Another goal is to curate genes that are associated to hearing loss and deafness using the gene curation framework. And then to distribute this information and these curations through ClinVar, through ClinGen's website, and through publications. So I figured I would start off by just going through a couple of background slides for hearing loss and deafness, just to start off with terminology. Usually when we use the word hearing loss, we're referring to hearing that's tested audiometrically that's below normal hearing thresholds. And then for deafness, if there's a lowercase d, we're usually referring to a hearing loss that's in the severe to profound end of the spectrum that's tested audiometrically. And then deaf with a capital D is a term to describe individuals that are part of the deaf culture, which has its own language and social attributes. Another term to refer to people that have deafness or hearing loss are hard of hearing. Usually this refers to individuals who have some usable hearing, or they may use some form of oral language. Often people that are hard of hearing may use hearing aids or have a cochlear implant or 2 cochlear implants. And I just wanted to take a moment to acknowledge that we're discussing hearing loss and deafness through a medical lens, but for deaf people, deafness isn't considered an impairment or a disability. And I just wanted to start the conversation off that way, and also acknowledge that I'm not a member of the deaf community. And I think this is an area that we want to make sure that we're respectful of the deaf community, and also just continue to learn. So there's a lot of good resources that we look at when we are using different terms to refer to deaf and hard of hearing and hearing loss as well. And then just to give a really quick background just on like what we're talking about through a medical or genetics lens for hearing loss and deafness. In general, we usually talk about 3 different types of hearing loss, and genetic hearing loss usually falls into the sensory neural bucket. So if you look on this slide, it has a picture of the outer ear, the middle ear here, which is these little ear bones here, kind of in this like blue space. And then the cochlea, which is part of the inner ear. And so the sensory neural hearing loss occurs at the level of the cochlea, at the auditory nerve, or within the brain. And then a conductive hearing loss is really anything that's from like the middle ear or out to the external ear. So that could be malformations of these structures. It also can occur due to fluid in the middle ear, due to ear infections. Perforated eardrums can also cause a conductive hearing loss. Earwax or blockages in the ear canal, these can all cause conductive hearing loss. Some of these can be genetic, particularly some conditions that result in malformations of these structures. And then for a mixed hearing loss, it's kind of a combination of both a sensory neural and a conductive component. And thinking about the causes of hearing loss and deafness, most of the time we are thinking about like an early onset, childhood onset deafness. Hearing loss, the prevalence of hearing loss increases with age. Age is a factor that can cause a hearing loss. But when we talk about a prelingual hearing loss, so a hearing loss that occurs before language is acquired, usually the prevalence estimates are somewhere around like 1 in 500 up to 1 in 250. And when we take that group of children that have a hearing loss, we think in the vast majority, around, usually in the literature, you see between 70 or 80% cited. Those are thought to be due to genetic causes. And then if you break that down further, the majority, or 80%, are non-syndromic and 20% are syndromic forms. On the non-syndromic side, the majority are autosomal recessive, about 80%. And then somewhere around like 19 to 20% are autosomal dominant. And then the remaining group is mitochondrial, X-linked. There are some RNAs that have been associated with deafness as well. And then just some other examples for acquired or environmental causes of deafness. There are some, I already mentioned age. So like as if you look at the population as a whole, as you broaden the age groups, the prevalence of hearing loss increases. So age is a factor. There's a lot of other environmental factors or acquired forms, particularly for prelingual or early onset hearing loss or deafness. The Torches infections, especially congenital cytomegalovirus, is a cause of prelingual deafness. And then for the syndromic form, some of the more common conditions that we think of that sometimes can appear like they are actually non-syndromic are Pendred syndrome. And Pendred syndrome involves abnormal structures in the inner ear. So it's enlarged vestibular aqueducts is something that you can see on imaging of the inner ear. And then some individuals have thyroid dysfunction. Another example is Usher syndrome. So Usher syndrome often presents with an early onset deafness or congenital deafness, and then retinitis pigmentosa, or a form of blindness that develops usually later. Another syndromic form that we can often see is Waardenburg syndrome, and this is typically inherited in an autosomal dominant pattern, which is unique from the other two syndromes I mentioned. So just kind of a high level overview of like the conditions that we're thinking of when we're curating genes and variants for hearing loss and deafness. So I'm going to turn it over to Marina. We're going to start off by talking about our gene curation working group, and then I'm going to take it back over from Marina later to further discuss the variant curation work that we've been doing.
Great, thanks, Andrea. So the ClinGen CDWG was one of the original CDWGs that was started with the formation of ClinGen. And so this was established in 2016. As you'll hear through our talk, hearing loss is highly genetically heterogeneous, and so we actually have a very large group of members who join this expert panel as experts in different genes associated with hearing loss. And there's a nice map there of where all our members are located. So we have relatively good representation. And at this point in time, when we made the map, we had 60 members at 27 institutions across 6 continents. Next slide. So I'll talk about the gene curation expert panel and the work that we've done and some of the challenges and interesting, unique pieces of curating gene disease relationships for genetic hearing loss. Next slide. So as Andrea mentioned, a lot of the hearing loss that we're focusing on is sensorineural and non-syndromic hearing loss. And so we really, and because there are so many genes that are posed to be associated with hearing loss, we really wanted to work to prioritize this. This was all published in our gene curation paper in 2019. But in order to generate our gene list, to give us a starting point, we looked at the genetic testing registry, which lists all of the genetic tests that are created by CLIA labs in the US at least. And so we generated a gene list of genes that appeared on 2 or more hearing loss panels in the genetic testing registry. And then we wanted to identify the diseases that those genes were associated with in a pre-curation. And then really we were prioritizing those that were associated with non-syndromic hearing loss. And so those already would just automatically go to our curation list. But then if there were genes associated with 1 or more hearing loss syndromes, we would do a rapid syndrome curation, which is kind of part of the pre-curation, really asking the question, is hearing loss a presenting feature? How many individuals are identified or will display hearing loss? Kind of to try to get a sense of the penetrance. What is the type of hearing loss? Really prioritizing the sensorineural hearing loss. Because as some of you may know, highly syndromic conditions may have ear malformations, and hearing loss might be a minor feature of those just because of the malformations across the syndrome. So we really didn't feel like many of the times we had the expertise to curate those types of hearing loss. And so if hearing loss wasn't a presenting feature or was a very minor feature, we would exclude those genes, at least from our primary curation list, because we just felt like we didn't have the expertise and we had so many genes in the first place to be curating those. But if hearing loss was a presenting feature for a syndromic disease, we would add that to our list and curate it. Particularly if we thought that it could be construed as non-syndromic or confused as non-syndromic hearing loss. We wanted to make sure that those would be added to panels for non-syndromic hearing loss too. For example, for Usher syndrome, if hearing loss develops first and retinitis pigmentosa develops later, we didn't want to exclude those from apparently non-syndromic hearing loss panels because those patients would be missed if we did. So that's kind of why we did this kind of pre-review of the gene list. Next slide. So here's our classification distribution, and this is up to date, not from our paper. And as you'll see, because of the way that we prioritized our gene list, over half of our gene disease relationships, and these are 149 unique genes and 177 gene disease pairs, over 50% were definitive. And then a chunk of them moderate and strong, which actually there was still a decent chunk of them that were limited. And so we felt encouraged that we should be looking at these genes that were on at least 2 panels in the genetic testing registry, really to summarize the gene-disease relationship and the evidence for these. That to remind everyone, ACMG came out with a statement in 2020 that genes should be, at least for clinical panels, moderate or above to be put on the panels, for screening panels, definitive and strong. So we felt it was important to curate these limited gene-disease relationships and publish them to the ClinGen website. Next slide. And this is just a breakdown of syndromic versus non-syndromic. As Andrea said, much of the genetic hearing loss that we were looking at was non-syndromic, and so it makes sense that 60% or so were non-syndromic gene disease relationships. But as I mentioned in our prioritization, we also did prioritize those syndromes where hearing loss was a presenting feature. So that is why we had 40% syndromic genes too. Next slide. And unsurprisingly, when you separate the curations by inheritance pattern, the majority are autosomal recessive per the diagram that Andrea showed in the beginnings, about 60% or so, 35% autosomal dominant, a smaller percent X-linked, and mitochondrial inheritance too. Next slide. And these are the classifications based on the frequency of the NGS panels here. And so as you can see, those that are on 10 to 17 panels, the majority of those genes are definitive, which we felt good about, or strong or moderate. However, there are a chunk of them that in the light pink are limited, or even some that are disputed and refuted. As you go down into less panels here, you'll see that there are some definitive genes on less panels, so 6 to 9 or 0 to 5, but some of those were syndromic genes associated with syndrome. So maybe certain companies wouldn't be putting them on their non-syndromic panels, although we feel that they might be appropriate to be on non-syndromic panels. And I think I have an animation for this slide. Yeah, so in the 0 to 5s, where I boxed there, those are syndromic genes. And then the disputed refuted genes that are on many panels are actually these 2 genes here, and we classified them and published them to the website. Next. So just to talk about some considerations and challenges. So as I mentioned, hearing loss has high genetic heterogeneity and also in most cases, low phenotypic specificity. So we're looking at sensorineural hearing loss, often prelingual, often profound. And there are some cases that the hearing loss pattern is distinct or it's associated with a syndrome, but this can, make us a little bit more suspicious of variants in just a random candidate gene if other genes haven't been investigated. Next. So the majority of genes we curated were autosomal recessive, and consanguinity in populations within these gene disease relationships was high. So to be cautious, we in general have been pretty suspicious of homozygous missense variants. And so we often have implemented a downgrade for homozygous missense variants and biallelic variants identified in consanguineous families, particularly if they don't have any functional evidence, just to be cautious. And de novo observations are very uncommon and are scored with caution, particularly in the autosomal recessive inheritance. Next. And then with 174 genes that we curated, there is a high re-curation burden, particularly because our group was started in 2016. Next slide. So just to remind everyone, these are the gene re-curation policies, and we did have quite a chunk of limited genes here and moderate genes. We found within setting ClinGen policies that moderates are the ones that change most often with time. And so re-curating moderates is every 2 years after the last approval date, and the other ones are 3 years. And a GCEP can also reevaluate sooner if a publication comes to attention or per community request. Next slide. So these are just the re-curations that we've done so far. We've redone 43 gene disease pairs that have been published so far to the website. And at this point, the GCEP is in maintenance mode, and we are planning to have a publication to evaluate these re-curations. So these are actually the final classification of the re-curation. In this publication, we're hoping to show, and maybe in a later Biocurator working group call, like how they've changed over time. You'll see the majority of the curations are limited curations with moderate also being involved too, 11 moderates. And in my experience, while we presented the recurations, we've often found that particularly the limiteds do not change from the previous the original curation to the recuration. So it'll be interesting to kind of look at some of these trends because we have such a high recuration burden to maybe help to further develop ClinGen policies. And I think that's the last slide that I had, Andrea. Yeah.
Okay, thanks. Okay, so I'll talk about our variant curation group, and I'll walk through like the formation of the group and then give updates on our more recent activities. So these are everyone here is probably aware of these aims and have similar aims for their groups. Whoops. So we set out to specify the ACMG AMP guidelines with hearing loss particularly in mind for variant classification. And then after those specifications were set, we wanted to review the variants and provide expert classification, and then resolve discrepancies and also resolve variants of uncertain significance using internal data from other ClinVar submitters and other members of the VCEP that might have internal data from their labs. So our VCEP, just like our GCEP and really our whole CDWG, is comprised of people with multiple different backgrounds that have to do with that focus on deafness in some capacity. So we have physicians, clinical geneticists, and otolaryngologists, as well as neuro-otologists. We have clinical laboratory diagnosticians. We have people that do more bench research, and we also have genetic counselors in our group. Of course, we also have biocurators whose time and effort has been really valuable over the years. And all of the members of the group have some kind of focus on genetics of deafness. So this is a timeline of where our group started off. So the early planning of the VCEP started in I think the summer of 2016, and or is that right? I think it was in 2016 at some point. I think that maybe summer is wrong because we had the group defined by November of 2017. And then we developed the classification rules, which was completed in 2018. And then we piloted those also in 2018. And we were, we were approved, initially approved as an expert panel in 2018 as well. So very similar to our GCEP, we have a lot of challenges for variant curation. One is the high prevalence of prelingual early onset deafness, and also non-genetic phenocopies are definitely something to take into consideration. There are some really well-known high MAF variants that like founder variants that we've had to deal with. There's also some variants that are kind of like hypomorphic alleles, so they may result in normal or borderline normal or very mild hearing loss. Those can be sometimes hard to work with during variant curation. High levels of consanguinity, especially for gene discovery, because most of genetic deafness is autosomal recessive. A lot of the initial publications were done in populations with high rates of consanguinity, so there's a lot of homozygous variants out there. And then also genetic heterogeneity. There's tons of genes. There's also a lot of, in terms of expressivity, there's a lot of variability even within families. And we also have syndromic and non-syndromic presentations within the same gene. Occasionally hearing loss can be overlooked, or it can be more it could be more mild, or as I mentioned, it could be borderline normal. One example is the USH2A gene, which is a major contributor to deafness, particularly Usher syndrome. And variants in this gene have significant overlap with both Usher syndrome and, sorry for my dog in the background. Hopefully you can all still hear me. Okay, I lost my train of thought now. USH2A. So variants in this gene can have significant overlap with isolated retinitis pigmentosa. And so some individuals may have normal hearing with variants in this gene, or they may have a really subclinical hearing loss that isn't picked up until a much later age than what's been classically described for Usher syndrome. So that does present a challenge for us as a hearing loss focused group to do variant curations. And then some genes are associated with both autosomal dominant and autosomal recessive forms of inheritance. So we had a lot to think about when we were specifying the criteria. Okay, so I'll walk you through our progress over the years. As I mentioned, the expert panel was formed, so summer of 2016 was really the early planning of this group, and we just tried to think of anyone in the field that would be able to contribute in some way with their expertise. And then we worked through the criteria, thought about each one and how they may need to be specified for hearing loss and deafness. And this was completed in 2018. Once the criteria were specified, we piloted them to test them out with 51 variants, and I'll talk about that process in another slide or two. We gained ClinGen approval as an expert panel in 2018, and the genes that we were approved for this initial approval are down here. So short list of genes. And then once we completed that, we started curating variants. Once we had variants curated, we went back and reviewed the impact of the specification, worked to resolve conflicts, and also resolve variants of uncertain significance if we could. And then a little bit more recently, we've broadened the impact. So of March of 2022, we gained approval for criteria for 2 additional genes, the OTO-F gene and MYO15A. And then currently and in the future, the activities that we've been focusing on are discrepancy resolution, resolving VUSs, as I mentioned, a lot of re-curation activities. Since our group has been around for a while, we've been focusing on the variants that require re-curation. And then there's so many genes associated with deafness, there's always a possibility to develop more specifications for additional genes that will probably always be on our future directions list. So this is just a slide showing the grid from the ACMG AMP criteria. And while we were working through this process, I think this was also back in the early days, this was also in the early formation of the sequence variant interpretation working group in ClinGen. So some of the criteria, they maybe needed a little bit more specification, but it wasn't necessarily hearing loss specific. So some of those criteria is kind of around the same time that we were working through this were also further specified by the SVI. And highlighted in the purple box are the ones that we ended up really focusing on. So the population data criteria, BA1, BS1, PM2, those were a major focus for our group. And those probably, I would say, took up the most amount of time while we were specifying the criteria. But there's other ones too, And I have some slides to give some examples on how we approached specifying these. Okay, yeah, so one example for the approach that we took for specifying the minor allele frequencies. So for the BA1 criteria for autosomal recessive deafness, we took like a global prevalence approach, and we used a really conservative prevalence estimate of early onset deafness at 1 in 200. And then we worked with some of the members of the VCEP who looked at their internal data. And we also looked at the literature to determine the allelic heterogeneity based on the most common pathogenic variants that were observed in those data sets. This also, I think that at the time we had data from EXAC, but I don't think NOMAD was available yet. So the allelic heterogeneity was set at 7.2%. And I think this was based on the 35del-G mutation that's in the GJB2 gene. And then because we were considering all genes, all deafness, we used a genetic heterogeneity of 1 and an estimated penetrance of 100%. So we were hoping that the conservative prevalence estimate would kind of balance out maybe some of the other parts or uncertainties in the heterogeneity and penetrance. And we used the CardioDB calculator to come up with a minor allele threshold for BA1 using these numbers. And this is also described in the publication that we have for the variant curation specifications that we made. And then for dominant hearing loss and deafness for BA1, we took a similar approach. I think dominant deafness was also kind of challenging because the prevalence estimate for dominant deafness isn't as well understood, I'd say, as recessive estimates, because some dominant forms of deafness can happen postlingually, and so you could have adult onset or later onset of the deafness compared to typically for recessive, we usually think of like an earlier onset. So we looked at a large population study of prevalence of deafness across different age groups. And we also wanted to be conservative, so we used the age group for ages 1 through 49. And the prevalence estimate for that age group is 1 in 15. And then also conservatively assumed that half of that was genetic, which is probably much lower than that. So we landed at 1 in 30. And then we're also challenged. There's a lot of challenges for like penetrance and allelic data in the literature because of ascertainment bias. If you look at the pedigrees that are published, the families that present and are studied are probably more likely to have a higher penetrance than maybe some other families. So I would say that probably our penetrance estimate of 80% that we set for this is a little bit squishy. We tried to use the literature. We also used our expert panels expertise to set that number, and we also wanted to be conservative because if we went off of the literature, it probably would be higher. And then we also used internal data and a literature review to land at an allelic heterogeneity of 5%. So that's kind of how we worked through the minor allele frequency thresholds for BA1. And again, it's further described in our paper. And then, this is just a slide showing our final minor allele frequency thresholds for each one of the criteria. We have removed, based on guidance from ClinGen, PM2 supporting. So our old PM2 is now PM2 supporting, and that's the same for both dominant and recessive. Another thing to mention is, I think I had mentioned before, there are some really high frequency variants that are considered founder variants that wouldn't really fit into this frequency scheme. So we do have a separate list of those variants that kind of, this wouldn't really apply because they have a lot of other data supporting pathogenicity. Okay, another example of the specifications that we did for PP4, so that's the criteria, is a supporting criteria towards pathogenicity if the phenotype is highly specific for a gene. Non-syndromic or isolated deafness, it's way too heterogeneous to use this criteria, but there are some genes that have unique presentations, and pathogenic variants in those genes have high prevalence for causing that disorder. So we do have a short list of genes where if the individual reported in the literature or potentially an internal patient from one of our members or a lab that submits to ClinVar has this particular phenotype, that's shown in this column here, provided that it would meet the other like inheritance, that there's two variants present, we can use PP4, and that's just based on the detection rate for an individual with hearing loss or deafness and these additional features that are under the phenotype column. The detection rate is much higher if you test that gene in those individuals. So we were able, even though at first thought non-syndromic hearing loss, you might not be able to use this criteria, if there are additional features that match the syndromic presentation, we were able to get approval for these special specifications. And every little piece of evidence definitely counts. So, okay. Oh, and I would say like these phenotypes, I think it was easy to develop these because they're very well understood and known phenotypes for these genes. Okay, so once we specified the criteria, we piloted those the new like specification guidelines that we are proposing, and to do this we had two curators from our group do dual curations for the same variant and make sure that they landed at using the same criteria and that they landed at the same final classification. And by doing this process, we were able to identify like some areas that maybe needed more clarity. And once the two curators had done this, we'd review this all on a conference call and make sure that there is consensus for the classification. So the first call for this process was in November of 2017, and then the final variant pilot call was in the spring of 2018. And we did this process for 51 variants. And this is just the slide showing the genes, the gene breakdown that we looked at for like what genes the variants were in. And then we also looked at whether the classification changed based on the final expert review. So this included like the work that the curator had done as well. So you can see here the like highlighted colors are showing if a variant originally was likely benign, and then it got pushed down to benign. That's like an example of how this chart is working. So a lot of variants were able to either get moved up or get moved down towards either pathogenic or benign using the new specifications. And then this was work from one of our prior coordinators, Mayor Patel. So he worked on this project to look at uncertain significant variants and also look at conflict resolutions. And he looked at 157 variants in ClinVar that either had conflicting interpretations or were uncertain significance. And after a review using the specifications, he found that about a quarter and almost 70% of VUSs were resolved either towards benign, likely path, or path, and 70% had an unambiguous classification. And then just to note, to date, our expert group panel has 185 variant submissions that are currently in ClinVar. So more recently, similar to the gene curation group, because our VCEP has been around for a while, we have had a lot of variants that we need to go back and recurate. I also already mentioned that we have new specification and approval for the OTO-F and MYO15A genes. So we started doing curation work for variants in those genes as well. But yeah, primarily I'd say for the last 6 months, we've been mostly focused on recurating variants that fit into the policy scheme for variants of mostly variants of uncertain significance, but also likely pathogenic variants that maybe could be moved up to pathogenic. And since we did the initial piloting of our rules, our group has changed in the way that we operate in terms of curation work. So I figured I'd just spend a minute going through how our VCEP process works. So we have a prioritized list of variants that we maintain, and then the biocurators in our VCEP will do a curation for one variant. So one biocurator does a single curation, and then we present those curations to the entire VCEP on a conference call. That happens once a month, and it's an hour-long call. Sometimes based on feedback or questions that the VCEP as a whole has, sometimes we have to go back to the curation step and do some follow-up work, and then before we can land at a final classification from the VCEP. But once there's agreement within the VCEP and amongst the experts and the chairs, we'll write a summary with the criteria, the classification, a summary text, and we make sure that that is reviewed and approved by the chairs. And then once everything is all set, we have everything in the VCI and we have a summary, we'll submit those to ClinVar. In terms of the Biocurator time estimates, we have never really tracked our time, which probably would be a good exercise to do, but I estimated that it takes anywhere between 20 minutes to 2 hours per variant. And even just doing one variant, I'd say it does take a lot of Biocurator time, because in addition to doing the curation in the VCI, we need to make slides to share with the VCEP. We need to review the previous curation that's in ClinVar and look for any new information. And then we're also, the biocurators are also responsible for writing the initial draft of the summary. So all of those things take time. It also takes time to present that information. So even a minimum straightforward variant, I still think takes about 20, it takes at least 20 minutes to do. And then again, we have 1 hour monthly call. Right now, our calls are really split between doing presenting variant curations and gene re-curations. So we don't have an entire hour dedicated just to variant interpretation. And then we also have a 1 hour monthly call with the chairs to make sure that we're on track with the things we need to be doing, and also getting approval for the final written summaries. And then this was a recent example that I presented. So we have a small list of re-curations that we've done, but this was a variant that actually the re-curation did impact the classification. So I figured I would spend a minute or two just to walk everyone through it. So this is a truncating variant in the Myo6 gene. This gene is definitive for autosomal dominant non-syndromic deafness. Interestingly, there are many biallelic individuals in the literature that present with a more early onset severe presentation, so kind of like an autosomal recessive form, but loss of function is established as the mechanism for both autosomal dominant and the autosomal recessive or biallelic presentation. So the initial curation was in 2019, and we really only had that it was a truncating variant in a gene that we know loss of function is an established mechanism. It's expected to undergo nonsense mediated decay, and it met our rarity or PM2 criteria. So it was classified as likely pathogenic in that time. And then very recently, in December, we looked at it again, and there were 2 new publications with 2 probands reported. And so since this is a dominantly curated gene, we were able to award PS4 supporting. And then one of the papers also had segregation information, so we could apply a supporting level of the segregation criteria, or PP1 supporting. And with all of that, it was enough to classify this variant as pathogenic. So that was rewarding to go through that exercise and move a variant from likely path to pathogenic. So I think that was, and yeah, just to wrap it up, future activities. So one thing, once we have a larger set of variants that have been re-curated, just as we're planning to do for the VCEP, it would be really nice to see what the impact has been of spending the time to go back and do these re-curations to see if variants are either upgraded or downgraded, just to see where they land. And then we will continue to work through our re-curations, both for genes and variants, curate newly identified genes as they're discovered, resolve discrepancies as much as we can. And then there's always opportunity to further specify and further improve our variant interpretation criteria for an expanded list. And also, there's always an opportunity to go back and look at the work that we've done to see if we can improve on it as well. And yeah, with that, I just wanted to take a moment to say thank you to all of the biocurators who have spent a lot of time doing curations for our CDWG, also the members and experts that call in through the years and give their expertise so that we can keep this group going. And then I just have our contact information there. So thank you.
Thanks so much, Andrea, and thanks for presenting with me. So it seems like we have a few minutes now for questions. So if anyone wants to unmute and ask questions, or also stick them in the chat, feel free to ask us questions and we can answer them for you.
I see there's,
Yeah, I
Sorry,
can go
I
over
was
the
just looking
one.
through the chat now. I didn't notice it while I was talking.
Oh, that's okay. I can go through the one question in the chat, which was regarding the gene curation process. Are there any differences between the variant curation process among different ancestries? And I answered that with the thing that we are most concerned. So we want to try to set thresholds for minor allele frequency cutoffs. So we just want to be as cautious as possible because we know that there aren't great population data sets for many of the populations where the hearing loss variants have been identified. So for example, in Middle Eastern populations, sometimes we have difficulty with population data sets there. So we really have to be cautious with determining their pathogenicity because a variant could show up in cases a bunch of times in the homozygous setting, for example, and it actually could just be common in that population, but we don't have population data to be able to tell us that. So if we case count over and over and over again, within a certain population, we could end up with a pathogenic variant that is spuriously pathogenic. So I think that's something that we are always thinking about. And then Andrea also talked about we would have a very different minor allele frequency cutoff for benignness if we included founder variants in that. So we do have a BA1, BS1 exclusion list so that we were able to keep our minor allele frequency cutoffs for benignness a bit more conservative at this point in time. But we also are always concerned that there might be other founder variants that we might have that are very frequent in a population we haven't had population data for example, and we might accidentally classify those as benign. So that's always something to consider. Any other thoughts on that question, Andrea?
No, I don't think so. I think you covered it.
So to follow up on the question above, has it happened that you would deal with founder haplotypes? Do you want to talk about how we've dealt with founder variants, Andrea? I'm thinking both of like autosomal recessive kind of case control analysis that we've done, and also I guess we can reiterate a little bit of the minor allele frequency cutoff stuff if that would help.
Yeah, I think that this is still challenging to deal with the high minor allele frequency variants, especially the ones that we think are hypomorphic that can present with a mild deafness or even like borderline normal. We've used like a case control approach to look at in population to compare populations that have been tested for genetic deafness that have the variant, so we know that they're affected, compared to either like carrier data sets or the like nomad data as well. So that's the approach that we've tried to take for those really high frequency variants, because you really can't use you can't really count cases in those scenarios. We also can use segregation information as well for those. So those are the types of data pieces that I think are the most helpful to try to resolve the really high frequency or founder variants.
And I'll add for some variants that we're starting to see relatively frequently in a current patient or certain patient population, we often feel better if we see that variant with multiple rare variant in trans with multiple rare variants instead of an additional founder variant, because we feel like it's much more common if another founder variant is common in the population to see it multiple times with this potential new variant. So that's another thing that we are cautious about. Any other questions? Okay, if not, we can let you all go. And thank you so much to Andrea for presenting this with me and, being a partner in crime for the last 7 years in the hearing loss expert panel. And thank you to everyone for listening, and happy 2023. And we look forward to many stimulating discussions in the Biocurator working group this year. Thanks.
Thank you.
Thanks, Vanda. Thanks, Marina. Stop share. There we go.